Okay, this is our in-class review that we did for our test on gene regulation and genetic engineering. So, first thing, in an electrophoresis, DNA has a blank charge and travels towards the blank end of the chamber. Remember, opposites attract. DNA has a negative charge because of all the phosphates. Phosphates along the side have this formula. There's a lot of negatives on there. So DNA is negative. Since it's negative, that means it's going to be attracted to the positive end. So C, DNA has a negative charge, is attracted towards the positive end. Opposites attract. Secondly, you want to create a plasmid. We often include a gene for antibiotic resistance. The purpose of that is for screening. Because when we make a plasmid and we put it in bacteria, just because we expose our bacteria to the plasmid doesn't mean that every bacteria picked it up. It would be like me throwing candy out to the room. Some people might grab a bunch of candy and other people might not get any. So we're going to have a mix of bacteria in here. Some of them picked up the plasmid, some of them didn't. So by having an antibiotic resistance gene, we'll know which bacteria picked up our plasmid. We'll just grow them in the antibiotic. So like in our lab, we grew them in ampicillin. If they did not pick up the plasmid, the ampicillin would kill them. If they had the plasmid, then they would be able to grow. Therefore, we screened them and we were able to get a pure strain. So that's the purpose of that. All right, next. They have, okay, so they're saying A and B are representing parents, and they want to know which of those could be children of those parents. To keep it simple, all you're looking for here would be that any child has to have a band that came from a parent. In other words, C, okay, if this is mom and dad here, C, this band is fine because both parents have it. This band is fine because dad has it. This band is fine, but this band is not fine because neither parent has it. So C cannot be their child. If you check the other ones, you'll see that actually D, E, and F could actually all be their children. So the answer here is D and E because F isn't an option. And again, you can double check this as long as any band that they have is found in at least one parent. If it's a crime scene, that's different. They need to match the crime scene exactly. But if it's paternity, they just have to have, a parent has to have a band if a child has it. Okay, simple vocab. DNA is cut with restriction enzymes. It makes sticky ends, and then we seal it up with ligase. So we cut it with restriction enzymes, it makes sticky ends, we seal it with ligase. This is simple vocabulary, you should know this from the lab. To make multiple copies, we use polymerase chain reaction, that's also called PCR. All you have to know about PCR, you don't have to know a bunch of detail. All you need to know is that it uses an enzyme, a polymerase. It's actually called Taq polymerase. It comes from a hot springs bacteria. The way it works is every time you heat up this sample of DNA, we get a copy because that activates the enzyme, and then you cool it down and that deactivates the enzyme. And so if you start with, let's say, 2 pieces of DNA, then it's going to be 4, then it's going to be 8, then it's going to be 16, and by the next day you're going to have 1 million copies of that DNA sample. And so that's how PCR works. Very, very basic how it works. When a bacteria is competent is just the word for making a bacteria transformable. Basically, it means that you've treated it with something, or the bacteria may be naturally competent, but its cell walls are weak enough that it will take in a plasmid. And that's what transformation is. In the lab, we made bacteria competent ourselves using a heat shock method. We treated them with a chemical, which was actually calcium chloride. You don't have to know that. We put them on ice, then we put them in a hot water bath, then we put them on ice again, and then we gave them food and we incubated them. These steps made them competent, and then we hoped they would pick up our plasmid, which everybody had good results, so it worked. Okay, which of these would create sticky ends? So the idea of restriction enzymes is that they recognize a particular sequence. In order for them to work, it has to be a palindrome. A palindrome would be a word that's spelled the same forwards and backwards. But in this case, since it's always going to read 5' to 3', it doesn't have to be the same forwards and backwards on the same strand. It just needs to be forwards, basically reading on one side, and then it needs to read backward on the other. So if we look at A, this is a palindrome. It's recognizing AATT, which it would be AATT, and on the other side it would also be AATT. However, this would not make sticky ends, because if it cuts between the A and the T, it's going to cut here, it's going to cut here, and this is what's going to be called blunt ends. Anytime it's the same number of letters on each side of a cut, you're going to get blunt ends. This one recognizes GAATC, so that works, GAATC, but if we notice, the other side, it's not a palindrome. This is not GAATC, this is GATTC, so this one wouldn't cut at all. This is your correct answer, CCATGG, and it cuts between the C and the A. And then when you read it this way, same thing, we get a palindrome, and it cuts here, and basically you get a jagged cut, and that's what they're talking about with sticky ends. Normally it wouldn't only be 2 bases hanging off, but the idea is if we cut a DNA and we end up with this, we can put pretty much anything we want in this space as long as it has the same sticky ends. This one's not a palindrome either, TTTTA, but when you read it the other way, it's TAAAA, so that can't be right. Okay, if we wanted to create a plasmid, this has got a bunch of steps. The question is which one of these are in the correct order. So in order to create a plasmid, the first thing we're going to need to do is get the original DNA out. So there's 2, which pretty much tells us that's probably the answer. Extract the original plasmid from the bacteria. So 3. Now we've got the plasmid and we've got the piece of DNA. Our next choice is to hydrogen bond the plasmid. In other words, we cut open our plasmid, we get our piece of DNA, they will hydrogen bond together, then we're going to add ligase. Ligase will seal them up, and our last step would be to then put them into a bacteria. So it's going to be this one, 2, 3, 5, 4, 1. Okay, matching. Simple vocabulary, you make sure you know all of these vocabulary words. So our first one, seals up DNA, that's going to be DNA ligase. Ligase seals. Secondly, a DNA that's ready for transformation, just went over that one. That would be, where is that? Competent. Next, made by the hot springs bacteria and used for PCR. That is going to be Taq polymerase. If you see anything about enzyme, it ends in ACE, so that makes it easy to remember. Separates DNA fragments by size. That's gel electrophoresis. Keep in mind, I wouldn't have to say size. I could also say by length. I could say by the number of base pairs. All of those things would mean the same thing. The more base pairs, it's going to be longer, which means it's going to be bigger, which means it's going to travel slower. It's going to weigh more. So all of those would be true. Next, cuts DNA at target sites. That is our restriction enzymes. Amplifies DNA. Make sure you know what the word amplify means. Amplify means to make copies of, and that's PCR. Next, DNA from 2 sources. That is called transgenic, or actually that's called recombinant DNA, sorry. But you should know that an organism that contains DNA from 2 sources is called a transgenic organism. And finally, the bacterial DNA that's used for transformation, and that's going to be a plasmid. Because if we were to use a regular DNA for transformation, it would be too big. So plasmid is smaller, and so it's easy to use for transformation purposes. Okay, so this is a transformation experiment. Guarantee you will see something like this, probably on the short answer part. So they tell you that you are going to put this plasmid into a bacteria. So this is the plasmid. They're always going to tell you what genes are on it and what those genes do. So this one says it makes a blue pigment when galactose is present. This one gives it resistance to ampicillin. This is the ability to digest lactose, and this is a resistance to tetracycline. So if a bacteria picks up this plasmid, it would have all 4 of those abilities. So they want to know, so you treat your bacteria. Remember, not every bacteria is going to get a copy of the plasmid. So we want to know, if we grow these bacteria on a series of plates, what's going to grow? Will it be only the transformed, only the non-transformed, both, or both, but they'll look different from each other? Well, here's a hint. You're never going to have a case where only non-transformed are going to grow. Transforming them is never going to give them something that harms them. So chances are, no matter what you do, the non-transformed, that's not going to be the answer. So you can eliminate that altogether. Okay, regular media. Media means what they're grown on. Regular media, which we use Luria agar, we called it LB for Luria broth. That's just a simple food. Therefore, there's nothing screening it. Anything can grow. So that's C. All right, in our second plate, we added ampicillin. If we add ampicillin, then that would mean that that will kill any bacteria that didn't pick up our plasmid. So the answer would be A, only the ones that picked up our plasmid or the ones that were transformed will grow. Our third one is our regular media, but now we've added the sugar galactose. Galactose does not kill bacteria. It's not going to harm them, and there's nothing here in the media that it tells you would kill them. There's no antibiotics. So that means everything's going to grow. But since the ones that picked up the plasmid have a special ability, they will be blue when we grow them here. So they'll both grow, but they'll be different colors. This one, minimal media, that means there's no nutrients. So basically what they're telling you is that the only food available is lactose. We're assuming that only the ones that picked up the plasmid have gained that ability to digest lactose. Therefore, the other ones would starve to death. So it's got to be A. And finally, over here at the end, media with galactose and ampicillin. Well, ampicillin will kill any of the ones who did not pick up the plasmid. So this is going to also be A. The only thing is, this one is going to look different from the other two that were also A. In other words, this was A, and this was A. However, this one will be blue because of the presence of the galactose.